Hello class, this is Dr. Naji with another model on the cell. What you see here is a foam model, or actually that's what I call it. I call it a foam model. You see here the cytoplasm of the cell. You could see here the plasma membrane, and you could see here in the middle the nucleus. If you look at the nucleus, it's bound by a nuclear envelope, and on top of that nuclear envelope we have many nuclear pores that are important for communication between the nucleus and the rest of the cell. This dense structure inside the nucleus is the nucleolus, and this nucleolus is important in making ribosomes and ribosomal RNA important for cell protein synthesis. This dense structure over here is the chromatin material. These are the condensed form of DNA in a non-dividing cell. If you look around here, around the nucleus, you could see the ribosomes. These are the brown structures that are found on the top of this structure here. These are all collectively known as the rough endoplasmic reticulum. Since rough endoplasmic reticulum have those ribosomes on top of them, then we can say that these rough endoplasmic reticulum are important in protein synthesis. These ribosomes, sometimes you find them bound into the rough ER. Some other time you can find them free ribosomes, as you could see in this depiction. These one here do not have those brown ribosomes around them, so these are called smooth ER. You could see them right in here. You could see them right also here. These smooth ER are important in are important in lipid and carbohydrate synthesis. After making those protein or lipid, we have to send them out the cell, and we have to put them in a secretory vesicles. This is when this Golgi apparatus or Golgi body comes in place. This is how it is in three-dimensional way, and this one here is cut so that you can see inside it there is a lot of crystin. So the Golgi apparatus are the one that make those secretory vesicles through which we are going to send them outside the cell through the plasma membrane. These structure over here are the mitochondria. The mitochondria are important for making ATP or energy for the cell. These two structure over here are the centriole. Centriole are important, or both collectively are known as centrosome. These two centriole are important in making the cytoskeleton that you see within the cell, and also important in cell division. What you see over here, some secretory vesicles. So these one here are the lysosome. These are the one that contain digestive enzyme. These one here are the peroxisome. These are the one that contain oxidase, catalase, so that we can get rid of the cell of toxic form of oxygen. Lastly, I would like to point to you that these are depiction of the cytoskeleton, that you could see them around the cell. These are the one that maintain the shape of the cell, as you could see them from this image. You could see in the surface of this model, these extension out of the plasma membrane. These are basically increasing the surface area of the plasma membrane. These are called microvilli. Microvilli are important in absorption, and you could see them within the cells of the intestine. In the cells of the intestine, instead of having one surface for absorption, we can increase the surface area of absorption by having those microvilli.